I'd like to introduce our second speaker, who is Professor Pierluigi Nicotera, who is a renowned scientist and leading international expert in the field of neuronal cell death. And he was appointed the scientific director of the German Center for Neurodegenerative Disease in April 2009. He was trained in general medicine and cardiology at the University of Pavia, here in Italy. His research has been centered on the molecular mechanisms that lead to neuronal demise following chronic and acute insults. Loss of neuronal synaptic connections and apoptosis play central roles in neurodegenerative diseases. I invite Professor Nicotera to speak about a slightly different title from what appears in your program, Epigenomics in Aging. Please, the floor is yours.
Tony, dear Jean-Louis, dear colleagues, when I was approached by my colleague and friend George Cass a few months ago to come and talk to this symposium, I was somewhat hesitant, and not because I don't believe that there are strong influences on nutrition, diet, food, on the development of neurodegenerative diseases, but simply because I thought I wasn't the right person to talk to you about that. There are a number of very interesting epidemiological studies which highlight the possibilities. And then I thought a little bit more, and along the lines of doing some studies on epigenomics in particular, and how these regulate not only disease, but particularly the process of aging, I thought that perhaps this audience might have been more interested in this kind of topic, and so I took the liberty, and I hope you will forgive me for that, to change the title and the topic of the lecture. So why epigenomics? Well, food, of course, and diets and so on are all elements which change our way of living, and lifestyle, of course, is a very important phenomenon that has changed over the past century. If you simply look at the longevity of the populations from the end of the 19th century until now, you will discover that we have, and this is very well known, I suppose, to everybody, we have actually doubled the survival time of the life span. What it is amazing, it is not only that we have doubled the lifespan. Of course, we can defeat early onset disease, we don't have infections, hygienic conditions have improved. But what is actually amazing, it is that even as we live longer, even people with disease live longer. So which means we have a form of adaptation to the long lifespan. So if you take, for example, Down syndrome, and I think that Dr. Trosko will come back with one example on Down syndrome later on. If you take Down syndrome, you'll see that basically in the 80s, about 25 years was the average survival time of a patient with Down syndrome. And today, we have nearly doubled the lifespan of these patients, which, yes, it is a testimony to the improved treatments that we have, the better condition, but also the fact that these people, within a short period of time, they have to adapt to live twice as long as they are supposed to. So to be able to explain such a short-term adaptation, we cannot invoke genetic changes. The genome doesn't adapt very quickly. And it's also very clear that at an age which before was considered to be a terminal phase of life, now we have individuals who are actually pretty fit. And they live a very good life, which in comparison to what it was a century ago, it is quite remarkable. The genome cannot adapt so fast. Genomic adaptation takes place when you have lethal mutations in a fast way. But adaptation to lifestyle, it is very difficult to take place in such a short period of time. So the only possibility to understand how we can adapt to live longer, to live double the time, is epigenomics or epigenetic adaptation. So, of course, I mean, the Time has a very nice cover here saying, well, our future is not determined necessarily by our DNA. I would probably dispute a little bit that title, but nevertheless it's a combination of factors. It is clear we have defeated some diseases, so we can make people living longer with those diseases, but also we have to have an adaptation of our genome to the long-term conditions. So let's look through some of the mechanisms and some of the pros and cons of having a long life. Yes, perhaps we have an epigenomic adaptation to live longer. But what does that mean? It means simply that we increase the probability of having genomic changes, so to have DNA damage, for example. If we live twice as long, then the probability, of course, increases twice as much to have DNA mutations that may actually bring about disease. The interesting thing it is we're learning as we go ahead that mutations are not necessarily mutations that take place randomly within one gene or one particular portion of a gene, but mutations can occur randomly within one single cell, one single group of cells. In the case of neurons, for example, there is a remarkable article which has been published just a few weeks ago by Christopher Walsh, which tells us that mutations can take place in quiescent cells, so in cells which are not dividing, and they are retained within these cells as a result of the fact that they do not divide anymore. The mutations are not replicative mutations. That means they're not occurred during cell division, but they are mutations which are occurring during active transcriptional activity of the genome. So when the DNA unwinds to transcribe genes, then that is the time when the mutations take place, which paradoxically tells us that these mutations may actually take place during activity. At the same time, in neurons, because these are not replication dependent, can tell us the history of the particular neuron in terms of his life, and cannot be detected by normal genomic screens. So in other words, SNP analysis or so will never reveal us this type of alterations, which means that yes, we live longer, yes, epigenomic factors may modify the chromatin, but may also open us to be able to have other type of alterations than normally we wouldn't have seen in the past. And don't forget that it's very rare the possibility that we would have a hidden genomic damage present throughout our life that would become evident and apparent only in old age, because there is no selection for that. Evolution would have never selected for a mutation that is apparent at the age of 70 or 80 or 90. So there is an interplay of genetic and epigenetic factors. In this nice review from Anne Brunette and colleagues in Nature Reviews, you can see that there is a possibility that epigenomic changes, like chromatin rearrangement, may produce a chromatin which is slightly more unstable, and then, of course, increases the probability of having then a genomic lesion due to mutations. At the same time, as we get older, the probability of mutations increases. So the interplay between genomics and epigenomics can be very important in determining susceptibility to disease in our life. So what kind of epigenetic changes are important? Nutrition, of course, is extremely important. We have changes brought about by physical activity, exercise. We have a number of factors which are very important, but because the focus here is nutrition, I try to focus a little bit more on this particular modifier of epigenomics. It was known actually from the 30s that caloric restriction could actually extend lifespan. And a number of other studies have then taken place over the past decades. And I quote here this review from Fontana, Linda Partridge, and Longo in Science in 2010, where they list actually some of the effects of dietary restriction. You can see this is conserved through species. I intentionally left out primates, non-human primates and men, because of course the evidence there is a little bit still controversial. There are papers like, for example, this one, which was a Richard Weindruck's paper a few years ago, showing that if you keep non-human primates to a dietary restriction, caloric restrictions, you actually have a sort of a more juvenile phenotype in the monkeys. On the right, for example, you can see the face expression of the caloric-restricted monkey doesn't look too unhappy to me, and also his fur is actually pretty good. Yet there were other studies which were actually saying, well, it's not true, it's actually different. We just save them from disease, but we don't make them living longer necessarily. So the issue is still open, but it is pretty clear that caloric restriction is one of the modifiers of the aging pattern. And of course, it is a modifier of disease. This is a very simple slide because I said I'm not the right person to discuss the effect of nutrition and neurodegeneration. There is this nice review from Bruno Velas and his colleagues that actually has a long table, a very, very long table with all the studies which have been doing or are ongoing on the effect of dietary restriction or modified diets on the onset of neurodegenerative diseases or their progression. Particularly Christine Jaffe has done recently a very nice study that I would suggest everybody to look at. But why nutrition, or what is the mechanism by which nutrition would actually affect our life? And is actually necessarily nutrition or rather metabolism or metabolic changes? So if we look at this figure from Andrew Dillon, review in Nature Cell Biology this year, he has actually quite a nice hypothesis. This hypothesis is a sort of a development of an old hypothesis from mitochondriologists. And the idea is the mitochondria age with a very limited lifespan. So because human beings are created to live in principle 50 years by the last century standards, before we had better hygienic condition and defeating diseases, mitochondria have to live 50 years. So the idea was that mitochondria themselves, and Doug Wallace was the one who proposed this initially, probably have a lifespan of about 50 years. And when you come at the age of 50, they start to be a bit sluggish. It's like an engine of an old car, and they don't metabolize anymore as they would do. They are slightly uncoupled in the first part of life, and they become sort of more coupled in the second part of life. So then there is a switch in the metabolism that is caused by mitochondria. Mitochondria tend to get a little bit bigger. There are defects in their clearance, and therefore we lose sources of energies. Now, the interesting thing that Andrew and his colleagues have done is that they have compared a mouse that is actually resistant to metabolic changes, which actually has an improved glucose homeostasis, with a wild-type mouse. And of course, those which are more resistant to the changes in metabolism live longer. And the others, of course, have an early death, or a normal death, if you wish, by the parameters in rodents. Now, if you really look at what happened, it is that the ratio between the consumption of glucose and the consumption of lipid, which is measured by the respiratory ratio here, in the blue line, which is the early, the wild-type mice, which die early. I think this pointer is dying on me. They have actually a very early change from the circadian difference in metabolism between glucose and lipids. So they tend to metabolize or to utilize lipids much earlier than other mice. Whereas in the green line, you can see that mice with an improved glucose homeostasis maintain the ratio for much longer. So the idea is that we tend to switch the metabolism from burning the glucose or the carbohydrates into trying to utilize lipids, which is what happened in middle age. And if we could stave this mechanism forward, then we would have an improved lifespan. Of course, what happened is that if you have an improved lifespan, then you develop at a certain time when the ratio goes down, anyway, metabolic diseases, neurodegenerative diseases, perhaps cancer, but you do that at a later stage. So what is the mechanism, and how can we manipulate lifespan? So I'm going to show you some unpublished work from my laboratory and Dr. Daniele Sbano laboratory in Bonn, where we actually looked at a particular model. This is a C. elegans, it's a worm, very easy to manipulate genetically, and therefore you can induce in this worm phenotype and genotype changes. And the things that we looked at in the past few years is the insulin stimulating insulin growth factor signaling pathway. In the insulin growth factor receptor in C. elegans is called DAF2. So if you make a mutant of DAF2, which means you block the insulin receptor signaling, these worms tend to live much longer. So the wild type lives about 21 to 30 days, the DAF2 lives up to 60 days. So it's actually double up the medium lifespan, so about 48, 49 days in the medium lifespan. So then if you decrease the insulin signaling, you reduce proteotoxicity also in neuronal model, you increase stress resistance, you promote tissue maintenance and neuronal function, and finally, you regulate metabolic adaptation. So what happened to the epigenetic and epigenomic regulation to these animals which survive long, which are DAF2 animals? Well, one of the things that we are looking at since a number of years, is the histone composition of the chromatin. Now, histones are very interesting because if you know the history of DNA, you know that back in the late 19th century, people still believed that the genomic information was kept by the histones. There was no concept of micromolecules. Friedrich Mischer then isolated DNA and basic proteins, and then finally, after the war, came back the idea that there could be strong, long macromolecules like the DNA that would contain the genomic information. So for a number of years, with the switch from basic to acidic material, people forgot about the histones. And they were considered to be some sort of structural component. In fact, we have an octamer of histones around each nucleosome, which is then wrapped around by 146 DNA base pairs. Now, these histones are quite peculiar because H1, H2, H3, and H4, they are expressed in a replicative manner. So in other words, when the cells divide, you need to have these histones, and so they are expressed in a replicative manner. But what happens when you have a cell which doesn't replicate anymore? So one of the things that happen is that one form, which is an isoform called H3.3, starts to progressively be replacing the other histones. So during our lifespan, there is a potential accumulation of H3.3. And this was a working hypothesis that we and others were working on that actually would bring to aberrant accumulation of H3.3 in the chromatin. And if H3.3 is accumulated, then what happened to gene expression? That's the question. And just very recently, again, this is a paper which I would suggest you to look at it. David Harris and his colleagues have been able to demonstrate that in neurons, during lifetime, there is a progressive replacement of the other histones by H3.3, because H3.3 is expressed continuously. The others are expressed only with the cell cycle. These cells do not divide, they accumulate H3.3. It is interesting that as long as you accumulate H3.3, but you destroy H3.3 by the proteasome, then you maintain a healthy turnover of the H3.3, which means that when you come at old age, your learning and memory are actually dependent very much on the maintenance of turnover of H3.3. This is true in humans and in other organisms, according to this paper. If you basically accumulate H3.3 without turning it around, then you have negative effects. So the question is, what happens if we actually delete H3.3? And that's what we have been trying to address recently. So if you take post-mitotic cells, and you assume that during life you accumulate H3.3, then you create a mutant, actually a deletion of H3.3, which we call H3.3IF. In these conditions, we may expect three effects. The first one, no effect. So basically, there is other isoforms which are taking place and which are replacing this. Number two is that we have histones which have the accumulation of the mutated deleted form. And number three, actually, is that the chromatin cannot arrange itself and you have loss of nucleosomes. And actually, I can anticipate to you that most likely is hypothesis number three, which is true in a number of cases. So you lose nucleosomes. You maintain nucleosomes which are functional, but you progressively lose others. So if you do a genetic screen and you try to find out what happens if you express in C. elegans in a insulin growth factor DAF2 model, which you remember has extended longevity, a mutation of the histone 3.3, you'll see that basically this is the normal DAF2 on the left side, the DAF2 gene expression pattern. If you express at the same time DAF2 plus the two isoform of histone H3.3 with the deletion, so effectively this one is a H3.3 knockout, you'll see that you completely change the pattern of expression into the worm. You end up having 8,220 genes which are significantly dysregulated in this model. So effectively, more than half of the whole genome. This is a volcano plot here on the bottom which shows you this. So 40% of the genome is dysregulated. It's a different regulation. It's a massive effect. So what happened to the survival of the worm? Well, if you take a wild-type worm, you will see that the displacement of H3.3 has no major effect. But if you take the one with extended survival, where we have altered the nutrition-dependent pathway, which is the DAF2 pathway, and the worm, which now lives much longer, the effect on the long-term survival, and I'm afraid that without pointer I can't show you the line, the effect of long-term survival, the black line is the control, basically, and the red one in the middle is the treatment with a mutant of H3.3, is decreased. So this animal now lives shorter. So which means that for a long-term survival, which is epigenetically driven, then you need to be able to have this histone H3.3 present to be able to mediate this effect. So this is an example of how nutrition-sensitive pathways are involved into extending lifespan, and at the same time that to be able to have this lifespan, one mechanism is the maintenance of the histone H3.3. How does H3.3 loads on the chromatin? H3 loads on the chromatins in a calcium-dependent manner. This is a study which we did together with David Michaud and Paolo Salomoni. And we demonstrated, this was a few years ago, that when calcium or activity-dependent neurons are, activity in neurons is stimulated by calcium entry, there is a protein called DAX. DAX, it is dephosphorylated by a phosphatase called calcineurin. Calcineurin then loads up the H33 and brings it to the nucleus and loads it onto the chromatin. Now remember what I mentioned before, that during activity-dependent transcription, there's a high probability of having mutations. So now it is a quite a neat mechanism that brings together both epigenomic and genomic regulation. Calcium is usually activated, or calcium entry into the neuron is active during memory or learning. You have a transcription-dependent loading of H33 onto the chromatin. So unless you clear up continuously that H33 by proteolytic cleavage, then what happens is that you have an unwound chromatin, which is much more a risk for mutations. One of the things that happen in aging, unfortunately, it is that the proteolytic systems are not as effective anymore. So this may lead to an accumulation of H33 and to a generally more relaxed chromatin, even during physiological processes, which may increase the probability of damage. So in conclusion of this part, before I go to the last part of my talk, modification of histones are much more important than we believe. They can be brought about by a physiological change and a replacement in aging. It is also remarkable that in the case of H33, these are not the only modification. I talked to you about learning and memory and accumulation of H33, but there are early changes of H33 which happen, for example, by mutation at the K27 position, which is absolutely important for development of glioblastoma and neuroblastoma for tumors. And I switch now to another topic, another form of epigenetic regulation, which is not based on the histone, but on the microRNAs. A few years ago, we stumbled on a particularly interesting mechanism by which learning and memory would be triggered or regulated by a set of miRs, or microRNAs, in particularly miR29. And this actually would take place as a part of a mechanism for addiction by nicotine or other agents working in brain as addictive agents. So we looked at the miR and the effect of the miR, and we concluded that miR29 was the most interesting among all of those. We studied this a little bit more in detail, and it turned out that if you knock down miR29, what happens is that neurons cannot form effectively synapses. So in other words, the number of synapses is there, but the shape of the synapses is different. They become more philopodia rather than mushrooms, and they are not able to have a normal electrophysiological activity. So in other words, there is a decreased activity. So why does miR29 absence produce this effect? Well, most likely, miR29 works as a brake, that when neurons are trying to form spikes around, it tries to stop them and say, hey, wait a minute. If you put too many leaves out, then the leaves are not going to be developing very well. They will remain like little philopodia. You have to let them grow into big leaves. So my at that time 6-year-old daughter, Julia, decided to make a drawing to express this. I'll go back to that one, because I was basically talking about that because she always tried to figure out what Daddy does. And I told her, I said, well, I work on these kind of things. And she said, but Daddy, you always go to meetings. And I said, yes, I have a lot of meetings. And do they pay you for that? I said, well, actually, yes. And so I was trying to get her more interested in the things I was doing. So I helped her to make this drawing to see that basically, if you make too many leaves on the trees, you have to have something to stop those, and that would be miR29. So that's the story which brought us to the miR29. So trying to understand how miR29 was working in plasticity. Well, guess what we found? Something completely unexpected. We generated a transgenic animal, which is a miR29 conditional deficient mouse, and it turns out that this mouse has a premature aging. It actually dies in 9 months. It has all the characteristic phenotype of aging, including kyphosis, including loss of fur, including cataract. So these are the survival curves. This is the eye defect, the kyphosis, the fur quality. So these months are actually living half of their lifespan. The interesting thing is that they have a deficiency in the collagen, which is associated also with aging. They have some metabolic changes, which it's be too early to discuss. Interestingly also, they have some hints or indication that their telomeres tends to be shorter, which in rodents is actually quite a remarkable phenotype. So why am I telling you about the miR29 knockout mice? Well, because another nutrient-dependent pathway, which is associated with autophagy and is the mTOR pathway, is primarily altered in these mice. So one of the reasons why they may age so fast is because in addition to the insulin growth factor pathway, the downstream events are converging onto AKT and the mTOR pathway, and in these animals, this pathway is activated, which would be, in a way, accelerating agents. There are agents like rapamycin, for example, that block the mTOR pathway, which are prolonging aging and survival in different species. So, to conclude, the aging process is very complicated. This is a figure from a review from Guido Kremer and colleagues. You can see here that there are a number of different components, not least the regulated nutrient sensing, which is the IGF-1 AKT pathway. And of course, these factors will cooperate together to be able to produce aging. The conclusion from these studies, or from these things I told you, is that there are definitely two very important issues there. We have to pay the price for aging. The price we pay is an increased susceptibility to damage. Yes, we age long, and if we age long, it's because probably there is a very important or a series of important mechanisms which have to do with nutrient sensing, with type of physical activity, with improved quality of life, which is an epigenetic mechanism. And this epigenetic mechanism on the other side will increase your sensitivity, our sensitivity to damage of the genome. So the whole thing, it is a balance of. So it's not surprising that we see diseases like neurodegenerative diseases coming very strongly at a certain age and old age. If you look at the GWAS studies, which have been done over the years, they really never came with anything surprising. Not to say they're not useful, they are very useful, but the genes which we discovered were housekeeping genes, genes which are regulating synaptic activity, inflammation, lipid metabolism, and so on in neurodegenerative diseases. So this tells us gives us a very important messages. These diseases are, in a way, unavoidable because we are running the car not for 100, but 200 or 300 or 400,000 kilometers. So they come to a certain point in which the only thing we can do to be able to prevent them is to understand which kind of epigenetic changes we can accept by reducing then the consequent damage which comes from this. And I would like to leave it here. In my last slide, I have the list of the collaborators that have been involved in this work, primarily Daniele Bano, our group in Bonn. Alessandro Fornarelli and Anastasia Pianova have done the miR29 work. Antonia Piazzesi and Drazen Panic, their work with the IGF-2, the DAF2 mutants. And in the past, Eugenio Fava, Ken Yang, Giordano Lippi, and Elena Ziviani, and Paolo Salomoni and David Michaud at UCLA College in London. And I would like to thank them and you for your attention.
Thank you very much. I now open it up to the floor. Please wave your hands visibly so that the microphones can home in on you. I think I've got one over there. Have you got a microphone? Can I get a microphone to somebody over there? What? No microphone, no hand. Is there anybody else?
Change your mind.
Yes, all right, please, Jean-Luc.
Thank you for your presentation. Would you be aware of any human trial showing that manipulating some micronutrients, like, for instance, folates, could have direct effects on methylation, for instance?
Mm-hmm. Yes, absolutely. I mean, there is, as far as I know, most of the trials which are going now, at least for Alzheimer's disease, of course, are, retrospective trials because you don't know exactly. There are no preventive trials for this, which would take years to be able to do. I know that there are studies where they are try to change nutrition to provide more elements for methylation or acetylation of the DNA, which would be a genetic regulation. But I'm not aware of any of them which is basically coming up with definitive data. Also because the problem is that you have probably to wait a number of years until you see whether there is a change in disease or in lifestyle or in sensitivity to inflammation, for example. It is pretty clear that at least in neurodegenerative diseases there is a convergence now of two important factors. One is inflammation, and particularly the role of the innate immune system. So in the microglia per se, the innate immune system, meaning, for example, the inflammasome and components which are actually dependent, as I was discussing before with Anne, from a generic state of chronic inflammation, cold inflammation. This is becoming very, very important. And the microglia itself as a mean to be able to spread the disease throughout the brain. So these are two key issues. So I'm convinced that in the future we'll see more and more of these studies which will highlight the link between epigenomic changes and the onset of disease.
Thank you. Are there any other questions from the floor? Another one? Go on.
Just one short question. When we talk about lifespan, increase or decrease, I'm always feeling uneasy because there are so many factors which can be involved.
Yes, absolutely. I mean, as I said in the last slide, I think there are multiple factors, so there is no one single factor. I wish it would be that way. We would be too correct. There are some experiments, though, which are actually quite intriguing, although very much debated. These are experiments from Tonis Viscore and colleagues, but also from others, where they created Siamese mice, so in other words, symbiotic mice. They take an old mouse and they cross its circulation with a younger one, so they put them together to live for a number of days or even months, and then what happened to this, it is that in the young mouse there are signs of aging, and in the old mouse there are signs of rejuvenation. So now, likewise, this seems to be, last century type of science. Nevertheless, he raised a lot of questions. These studies raised a lot of questions from the community, so they're trying to figure out what kind of patterns really is important to be able to create age-related effects in these model systems.
Thank you. There's a question up there.
Yes, hello. Are there any diseases known to be associated with a mutation in the histone H3.3 gene?
Not that I know. There are mutations which are associated with mutations of other histone, H3.3. As far as I know, I don't know any that has been found so far.
Okay, thanks.
And also, because I think I wouldn't expect necessarily a disease, because if you saw the data I showed, this H3.3 in principle does not have any effect on wild-type worms. The effect is only when you have an extended lifespan. So he probably has an effect at the time in which it starts to be accumulated. So if you prevent that accumulation, which is supposed to happen according to the papers I showed you earlier during the aging time, then you have a problem. So you need it for the extended survival, not for the early life.
Thank you. Thank you very much. I have one more question, last question, near the front, third row. Thank you.
So the prenatal effect of an unfavorable environment, would this in a way affect the mechanism that you have described?
Yeah, I think that's a very important question. I mean, it's at different level. So the one I was describing for H3.3 definitely would be a later life effect, but there is a school of thought, a number of people who think that the perinatal period as a whole is the most important for epigenomic regulation. So that during the prenatal period, but only during delivery and in the first months after birth, that the modification of your epigenomic imprinting is very, very important. I'll tell you about another study which has been published by one of our scientists in Bonn. They have mated old mice with young dames and compare the mating with the young mice and young dames. And then look at the offspring, and it turns out the offspring has this a bit of the old guys with the young ladies. They have a impaired learning and memory. So this would indicate that you have an epigenomic imprinting in the sperm that then is transmitted to the next generation. So if this happens at the time of the very, very beginning of the formation of the organism, I'm sure that there are a number of other changes which then will influence during the in utero life, but also afterwards, in the first months of age, the epigenomic imprinting, if you want.
Right, well, thank you very much, Pierluigi. I'm very conscious that we've all aged 40 minutes during that, but it was a fascinating contribution. Thank you very much. We now have a coffee break. Please, could you all be back here in the auditorium at 4:20, and then we'll start again. And thank you for those online who are listening. We reconvene at 16:20. Thank you. That was great. Good. No, that was...